maybe we can, let's get started here. I just wanted to start by saying a few words to the QBI and QCRG community about the current state of affairs in the world. Obviously, we are facing tremendous challenges, not only with the pandemic, but importantly with the social response to many recent and unacceptable actions that have taken the lives of our Black citizens. I feel hopeful that these protests that are ongoing ultimately will bring about overdue significant change to a really unjust system. And I just wanted to share a couple of recent statistics with you that I was recently made aware of. 1 in 1,600 Black people have died from COVID-19 in this country to date, which, by the way, is 2.3 times higher than the death rate for white people. However, it is still not as high as the likelihood of a Black man being killed by a police officer during his lifetime, which is 1 in 1,000. So at QBI, we are trying to put together a plan to address some of the inequality and underrepresentation of Black students, faculty, and other colleagues. And I would like to stress that we do not think we have any solution by ourselves, and therefore I am asking you to please contact us with your ideas towards what you think we can implement for lasting change. So any ideas that you have, please contact myself and or Jacqueline. Okay, I'll share my screen now, Alexa. So welcome to the second town hall from the QBI Coronavirus Research Group, the QCRG. In the first town hall, we had 4 subgroups represented. Brian and Klim and Cherie and Davide presented. And today we're going to have 4 other subgroups presenting their efforts. Melanie Ott with virology, Natalia and Eric on mitochondria, metabolism, and aging. Danica Fujimori is going to represent epigenetics and chromatin, and John Gross is going to talk about ubiquitin. And I just want to highlight 2 other subgroups that have been recently formed. Martin Catman here is heading up a genetics subgroup, and Bruce Conklin and Todd McDevitt, they're heading up a cardiovascular subgroup. So very excited about these 2 new subgroups, and if you're interested in being involved in these 2 groups, please contact these individuals. But today, the order is going to be Danica and then John Gross, and then Natalia and Eric, and followed by Melanie. And the last slide I have, I just want to advertise a symposium that's 1 week from today that we're having. It's a QCRG COVID-19 Research Symposium next a Friday morning, involving a number of speakers from QCRG, but many more from around the world, many of which we've been collaborating with over the last couple of months. And in the bottom left-hand corner here is a link for you to register. So please register and attend 1 week from today. So I'll stop sharing, and I'll remind all the speakers that you each have 10 minutes, and I'll remind the participants that any questions you have, please put them into the chat box, and we'll go through them at the end of the 4 talks. We'll try to get through as many questions time permitting. So Danica, if you wanted to go ahead.
Yep. Can you hear me okay and see my screen?
Yes.
Thank you for the opportunity to present work that's been going on in QCRG's epigenetics subgroup. It's great to have everyone on the call and hear questions and ideas that might arise. So in our subgroup, sorry, there we go. Our subgroup investigates host factors that have been identified as interacting partners of the novel coronavirus proteins and are typically associated with chromatin regulation and DNA replication. In addition, we also have interest in novel coronavirus microdomain protein. Currently, our subgroup has 50 members and a strong synergy with a number of other subgroups, including drug discovery, virology, bioinformatics and system bio, as well as structural biology, structural modeling, and many others. I will review in upcoming slides these epigenetic factors that have been identified and tell a bit more about all of them. Chromatin-associated factors called bromodomains have been identified as interacting partners of the viral envelope protein. or protein E. Protein E of the novel coronavirus is a small protein that is predicted transmembrane protein with localization in ER and Golgi membranes. While not the dominant envelope protein of the virus, protein E is involved in critical aspects of the budding and assembly in the viral life cycle, and is predicted to form a pentameric core with a putative ion transport function. Protein E is expressed at remarkably high levels in infected cells, and it's critical for productive infection. And lack of this protein strongly reduces viral titer and impairs viral maturation. Thus, virus-lacking protein E has been suggested as a potential vaccine candidate. Now, 2 human bromodomain proteins have been identified as interacting partners of the protein E. Bromodomain proteins, BRD2 and BRD4, contain bromodomain and a so-called ET domain, and are a family of epigenetic regulators that associate with chromatin by interacting with acetylated lysines. These BET proteins are critical co-activators of interferon response genes in the lung, and are involved in transcription elongation of interferon response genes. They are also activators of pro-inflammatory response associated with lung fibrosis, COPD, as well as asthma. So since bromodomains associate with chromatin as a function of lysine acetylation, the question that we were wondering about is whether there are any histone-looking lysine residues that might provide the landing pad for bromodomains within the E protein. And it turns out that the C-terminal segment of the E protein, which is highly conserved in coronaviruses, contains a close mimic of histone 3. Thus, the model is that acetylation of this conserved lysine residue within this C-terminal segment might provide a binding site for bromodomain, allowing for sequestration of bromodomain, and in that way, inhibition of their antiviral response. I've learned from Nevan yesterday, the genetics data is just coming in on these bromodomains, and we are quite excited to see what these genetic experiments will inform. Another epigenetic regulator identified as interacting partner of, in this case, NSP5 viral protein is a histone deacetylase enzyme HDAC2. As the name says, this is an enzyme that antagonizes lysine acetylation in histones. And HDAC2 in related contexts, it's shown to be a component of influenza A-induced host antiviral response. And transcriptional activity of this histone deacetylase regulates expression of antiviral genes, including viperin. HDAC2 regulates inflammatory responses, and especially in pulmonary contexts, which could be quite relevant to pathogenesis of COVID-19. Low expression of HDAC2 contributes to severity of COPD. It has been predicted based on the sequence of HDAC2 that nuclear localization signal of the protein is cleaved by NSP5, which is protease itself. In this model, protease would remove nuclear localization signal, and in that way prevent localization of HDAC2 within the nucleus and its transcriptional program. This model is under active investigation currently. DNMT1 is a third epigenetic factor identified as the interacting partner, in this case of ORF8, accessory viral protein. DNMT1 is a DNA methyltransferase enzyme, which has known evidence for role in viral infections. It has been shown to be required for Hep C infection. And importantly, clinical inhibitors of this DNA methyltransferase, such as chemotherapeutic agents azacitidine, are known to suppress HCV infection in cell culture models, suggesting potentially use of DNMT1 modulators as a potential therapeutic strategy for the disease. Finally, NSP1 viral protein, which antagonizes interferon induction and increases production of pro-inflammatory chemokines, interacts with the components of DNA polymerase alpha complex. And these are proteins shown in here. Now, DNA polymerase alpha complex regulates activation of type 1 interferon. And also in the context of coronavirus infectious bronchitis virus, related interaction in between a viral protein and components of a DNA polymerase complex induces DNA replicative stress. It is yet to be seen if that is the context, if that is the case in the context of the novel coronavirus. As I mentioned, in addition to these host proteins that interact with viral proteins, there is one viral target that is also of interest to members of the subgroup, and that is novel coronavirus's macrodomain protein. What are macrodomains? So these proteins antagonize ADP ribosylation of proteins, and ADP ribosylation is a large post-translational modification that adds two riboses and an adenine moiety to proteins. Now, this modification itself is introduced by ADP ribosyl transferases, which are also known as PARPs. And ADP, members of the PARPs family are induced by interferon response that are known to inhibit viral replication, presumably through modification of either host or viral proteins. Now, by antagonizing PARP activity and removing these modifications, macrodomains might prevent that an antiviral response that PARPs have. In the context of the original SARS virus, each macrodomain promotes virulence and suppresses innate immune response. That, together with the conservation of macrodomains across coronaviruses and their essentiality in pathogenesis, has led to a nomination of the macrodomain protein as a potential therapeutic target, and current efforts of the subgroup in collaboration with structural and drug discovery team members are focused on identification of small molecule inhibitors of macrodomains. And with that, I'd like to thank all the members of Epigenetics subgroup as well as the broader QCRG scientific community. And thank you all for listening.
Thanks, Danica, fantastic. We'll just keep going here. John, you're up next, talking about ubiquitination regulation and SARS-CoV-2 infection. And you're muted.
Okie dokie. Gotcha. All right, is everyone able to see title slide?
Looks good.
Okay. Let me just do some housekeeping here. I have to move. As much as I like looking at you, Nevan, I have to move you out of the way so I can see my slides. Okay. Right, I'm going to tell you about the ubiquitin subgroup. It's always a pleasure to speak in front of my colleagues. I wish I could see all of your faces. I miss you all and interacting. I wanted to start with just a perspective on RNA viruses and how they can, wreak such havoc with such a limited protein coding capacity. And many of you know this, but some of you may not, especially new students to the project. What I want to emphasize here is that these encoded proteins have to multitask, and so some of the encoded proteins can have multiple structural forms. It can be polymorphic. I think this is well documented in Ebola. These proteins also have to make interactions with host factors. Sometimes they're different host factor partners for different functions. So it's a challenge to try to define these things. And Nevan's lab over the years has come up with methodologies to look at this. You'll hear about those methodologies today. And we've been fortunate to be along. We're interested in this question of how viruses suppress innate immunity, which is a barrier for infection. And it's actually a barrier for cross-species transmission in primate lentiviruses. And we've been working with expert evolutionary biologists to kind of merge a structural biochemical lens with evolutionary biology to understand how that happens in the genesis of HIV-1. So I'm excited about that work. We just published something recently on that. And I guess I hope as part of this group, we can make some inroads in how SARS-CoV-2 hijacks ubiquitin to suppress innate immunity. So this is just a quick, my only HIV example, but I think it makes the point that, you know, HIV can spread in CD4 positive cells, but if there's an accessory protein that's knocked out, the virus just gets blocked in its tracks. And so this is a multi-log unit effect, and this is an example of innate immunity. As many of you know, what's causing all the green lights to go out is the ApoBec3 family of restriction factors. So this is very black and white genetics. There's a restriction factor that forms an innate immunity, and then there's a viral protein that shuts that down. Okay, so we're very interested in those kinds of interactions in RNA viruses. So that was a retrovirus. We're talking about an RNA virus that uses an RNA-dependent RNA polymerase to replicate, copy its genome. And such viruses are counteracted by the interferon pathway. And what happens is double-stranded RNA that's made by this RDRP is sensed by nucleic acid sensors, such as the toll-like receptors or RIG-I or MDA5. And there's a signaling cascade that propagates down through a critical kinase called TBK1, which phosphorylates a transcription factor. And this allows for the synthesis of interferon alpha and interferon beta. Those are secreted and can act in a paracrine or autocrine manner. They bind to the interferon receptor. And this leads to the induction of interferon stress genes. One of them is ApoBec3G, by the way. There are many others, PKR, which act as a potent defense against viruses. So this is a pathway, and one way we're looking at this large data set generated by Gordon and some of the other data sets. So here's the analogy to ApoBec. You can see here that SARS is very potent at suppressing the production of interferons. Okay, so, and there are several proteins, both non-structural proteins and ORFs that have been documented to be involved in interferon suppression. So the virus is using multiple ways, and you heard Danica talk about one of them through this macro domain. The virus is using its enzymes like the macro domain or ASP3 PPL Pro to suppress interferon, but also structural and ORFs as well. So going to great effort, how does it do it? I think that's a really rich question. So the way many viruses suppress innate immunity is by hijacking host ubiquitin pathways. And this is just a quick overview that one way the viruses hijack ubiquitin pathways to target an interferon restriction factor like ApoBec or others for degradation by the proteasome. Okay, so the virus will hijack a ubiquitin protein ligase, which adds polyubiquitin chains with a specific linkage to promote its degradation. But viruses can also intercept ubiquitin mediated signaling. Okay, so I just want to point that out because this interferon pathway uses a lot of ubiquitin signaling where you have K63 chains which don't act as target proteins for degradation, but instead nucleate protein complexes involved in this cascade I just mentioned. So here's the group, and we've got expertise from many different labs, from crystallography to systems biology to chemical biology to protein biochemistry and ubiquitin. So we're very pleased to have this team of people that comes together on a pretty regular basis. So there's a lot of data in the Gordon interaction map. So here's just a lens of innate immunity in terms of the viral proteins that interact with known E3 ligases involved in the production of interferon alpha and beta. So for example, NSP15 interacts with this E3 ligase called NRDP1. That's at the top of the screen. And then on the right is NSP9, which interacts with an E3 ligase, with the E3s are colored in blue, by the way, called MINBOM or MIB1. And then there's ORF3A, which interacts with TREM59. So I should add that NSP15 is known to act as an endonuclease for RNA to suppress innate immunity, but this is an intriguing connection because perhaps it also works to, has a double job, does a double whammy. Maybe it interacts with the ubiquitin E3 ligase to suppress signaling. So genetics, as Danica alluded to, genetics, RNA, these are really going to be critical to figure out where the money is, where is the function. But for now, we're just going to talk about the data and not be encumbered by any experiments. So let me tell you a little bit about, I'll show you a little experimental data at the end, I promise. So let me just tell you about MindBomb and NRDP1. Both of these E3 ligases put K63 chains on this kinase, this master regulator of interferon called TBK1. TBK1 is a kinase that's activated by K63 chains, and that promotes the phosphorylation of IRF3, which can dimerize, go to the nucleus, and make type 1 interferons like interferon 1 and alpha and beta. And so these are known to be antiviral E3 ligases, so it's tempting to speculate that the interactions of NSP9 and NSP15 with these E3 ligases may suppress their antiviral function. That's to be determined. The next interaction is one between an accessory protein and a Cullin E3 ligase. This is a well-studied superfamily of E3 that's actually hijacked by many viruses. And so there is a connection between ORF10 and Cullin-2. And there's a paper by Stephen Elledge that describes ORF10 Cull2 interaction as being required. Sorry, I'm rushing. There's a paper that describes the interaction of this protein called Zyg11B with Cull2. Zyg11B is a substrate specificity factor for Cull2 that promotes degradation of glycine N-terminal glycine degrons. And this has been involved in the clearance of fragments produced by caspase cleavage and also inappropriately in myristylated proteins. So maybe there's a connection between ORF10 and apoptosis or membrane quality control. Again, that remains to be seen. I should say, ORF10 is controversial. It's not clear that it's actually expressed. And we've been interacting a lot with members of the translation subgroup to try to figure out, is this ORF really worth going after or not? One thing I should say is that reports suggest, a preprint suggests that the pan-Cullin-RING Ligase inhibitor, pavondistat, actually inhibits SARS-CoV-2 infection. So to me, this suggests that a Cullin or multiple Cullins may be involved in the replication of SARS-CoV-2. So, we are going ahead and reconstituting some of these Cullin complexes, and this shows here just some progress making the substrate receptor of Zyg11B, LOB, and LOC. So that's by Shilu, it's a postdoc in my lab. He worked some related complexes from HIV, so this was straightforward. Next, what we want to do is something we've done before for other viruses, is work with Nevan Krogan's lab to identify the substrates of E3 ligases that could be hijacked by SARS-CoV-2. And in this experiment, we used a technique called ubiquitin remnant profiling. In this experiment, Viro E6 or A549 cells are infected with the virus, and then there's a treatment with MG132 or not. Cells are lysed, trypsinized, and the fragments that result contain a remnant from ubiquitin, which, as you know, the C-terminus of ubiquitin makes a covalent bond with the lysine. So they're antibodies specific for this GG remnant, which actually results from other ubiquitin-like proteins as well. So you get ubiquitin and other UBLs in this analysis. You can pull down and enrich for those peptides. And then you can couple that with protein abundance measurements to see which proteins are ubiquitinated and degraded during infection. And we can also do this, and Ruth is doing this right now for ORF10 or other single proteins stably expressed in cells. So this is some work. I'll just give you a little taste. Some new data just came in. And I'm going to mention just one. This is cherry picking, but I'm going to mention NSP9. This is the protein interaction data from David Gordon and the large groups effort. And you can see that MindBomb is actually enriched in ubiquitination. This doesn't say NSP9 is doing it, but it's tempting to speculate that that could be. the case. Some proteins are depleted for ubiquitin signatures. So this is work that was carried out in infection in the Vignusi lab, Marco's lab in Paris, and we're just keen to see more of this kind of analysis. I want to add this just for fun. DCAF7 happens to be an interaction part of NSP9, and that's the subunit. That's an adapter for another Cullin-RING ligase. So there could be multiple Cullins at play here. So we look forward to going deeper into this. We're just getting started. Obviously, things are going pretty slowly from where we're sitting based on the shutdown, but I really want to thank the whole consortium, specifically though interactions with the translation subgroup and the drug discovery subgroup and the structural biology subgroup. And I look forward to interacting with Martin on some of these genetics tools he's bringing to it. So thanks a lot.
Thanks, John. That was a really exciting presentation on the ubiquitination connection. It'll be great to see all these connections fleshed out in the near future. So we're going to keep going here and go to the next set of presenters. It's a tag team presentation from Natalia and Eric on metabolism, mitochondrial function. And I think Natalia will go first. And perhaps Natalia is muted.
Sorry about that. Can you see my screen?
Yes, looks great.
Okay, awesome. Welcome everyone. So I'm going to go ahead and get started and tell you, hold on, can I? Yes. So since last time, there has been a major change in our group. We have merged with metabolism subgroup head by Eric Verdun, who's gonna speak in the second half of this presentation. And we're truly bridging the Bay Area expertise here. I listed some regular members of our group who tune into our biweekly meetings, but we always welcome more. So if you guys are interested, I really want to encourage you to join. So the subgroup was inspired by the observation made by Nevan's lab that approximately 15% of all host proteins that associate with SARS-CoV-2 are either mitochondrial proteins or have something to do with mitochondrial proteins or mitochondrial pathways. And this is, of course, exciting because mitochondria are so central not only to energy homeostasis in our cells, but also to orchestrating innate immunity and cell survival under conditions of stress associated with pathogen infections. And mitochondria, of course, are not static organelles. They're very dynamic. They transit between this fist and fused states, and different states of mitochondria differentially impinge on these functions that this organelle has. And over the years, it has been shown for a number of viruses that they interject in these transitional processes in very different ways, and ultimately always hijacking the function of mitochondria for their own benefit. And I would argue that at the molecular level, how this really happens is not very well understood. And the protein-protein interactions we now have available through Nevan's work really allow us to sort of ask those questions for SARS-CoV-2. You know, we clustered all the human host proteins, which are of mitochondrial connection, into these functional groups, and you can see that we can sort of categorize them in different classes that have to do with different functions of mitochondria. And our back colleagues really focus on fatty and amino acid metabolism and aging part of it, whereas at UCSF we are interested in all these other processes. And I just want to highlight very briefly two connections that we're interested in right now in our subgroup before I hand it over to Eric. One of them centers on TOM70 that has been pulled down with ORF9b, the viral protein. It's a truly fascinating protein. It's a membrane mitochondrial import receptor that typically imports proteins into the mitochondria, but also it can associate with this MABS membrane protein that John has mentioned, which now interacts with RIG1, which is a sensor of viral RNA. And through this, TOM70 sort of participates in relay of information to the TBK1 STING pathway that John mentioned as well, and NF-kappa B pathway. And by interacting with TOM70, we think that ORF9b really has a potential to intersect at many different paths of this, or points of this innate immunity and adaptive immunity pathways. So we have Giotti from the Krogan group and Yanshin from the AGAD group who are looking at TOM70 from both biology and structural biology perspective. We're very interested in what is going to come out of this. There is a connection between MABS and SARS-CoV-1 that has been published years ago, but molecularly, we don't know how it works, and maybe TOM70 is the missing link. The other thing I would like to highlight is our discussions on how COVID-2 intersects with many different aspects of cell survival pathways that are associated with mitochondria, and in particular the necroptosis pathway. My lab is very interested in the fact that RIPK1 kinase was pulled down with NSP12. Seemingly, this is a very bizarre connection, and NSP12 is an RNA polymerase. But closer look on one of the domains of NSP12, neuron domain, made us realize that actually, and that's noted in the literature, that neuron domain looks like a kinase domain. It has features of kinases, and that's very interesting because activation of RIPK kinases in necroptosis are very much dependent on kinase-kinase interactions, oligomeric interactions. So we're trying to figure this out both via the structural QCRG consortium and in our lab, spearheaded by Raf, what it means biologically. So those are very short highlights. I want to finish with this slide just to place the members of our groups on our interactome, a mitochondrial interactome, host interactome, just to show you that, you know, we're covering some of it, but there is still a lot of notes which are exciting. So if anybody is interested and would like to follow up with us on this, please get in touch with me and Eric. And I'm going to now hand it over to Eric.
Oh, Eric, you're muted.
Yep. Okay. Yeah. Thank you, Natalia. Can you see my screen?
Yes.
Okay. Very good. So I will delighted to be here and to see all of you. I'm going to be discussing one small story that is emerging out of our collaboration with Natalia, and it's dealing with NSP14, which is a proofreading enzyme. It is in complex with another viral protein called NSP10. It's an extremely highly conserved protein, about 98% similarity with the orthologous protein from SARS-CoV-1. It has a 5' exonuclease proofreading function that ensures the high fidelity of RNA replication of the virus. Also has a methyltransferase capping activity, which is shown here on the side. So the structure of the enzyme from CoV1 has been solved, and it's shown here, the two different domains, the exonuclease and the methyltransferase activity. And shown here is the interaction with between NSP14 and NSP10, and the interaction is critical for NSP14 activity. So out of the Gordon paper came out of an interesting finding, which was that one of our favorite enzymes called SIRT1 interacted with NSP14, and so we've been busy trying to validate these results and explore their biological significance. So brief word about sirtuins. We've been working on them for 20 years. We cloned the mammalian equivalent of the sirtuins. There are 7 sirtuins. They have a characterized central catalytic domain, which is shown here conserved, and various N and C terminal extensions. All of the sirtuins are present in different subcellular compartments, nucleus, cytoplasm, mitochondria, and they are uniformly have been identified as protein deacylases. I'm showing you here one acyl group. It's an acetyl group on the epsilon amino terminal group of a lysine. This group is removed by the sirtuins, and this is done in cooperation with by cleaving NAD, which generates nicotinamide, and the ADP ribose serves as an acyl acceptor, which is shown here, generating in the case of an acetyl ADP ribose. It turns out SIRT5 is actually quite interesting because it is specialized for acidic acyl groups, and this is shown here. It targets malonyl lysines, succinyl lysines, and glutaryl lysines. We've been involved in working on SIRT5 for the past 10 years. We've published a number of papers here, which included a characterization of the first malonylome and the first succinylome, and trying to understand what the biology of these modifications are. Turns out there are they play a key role in numerous biosynthetic pathways, both in the cytoplasm, in the nucleus, and in the mitochondria. SIRT5 appears to be localized in each of these compartments. So the first thing we did was to validate the interaction, and this is shown here very briefly, showing that indeed if one overexpresses NSP14, you find SIRT5 co-immunoprecipitating with it. We repeated this with other sirtuins, and we're actually surprised that the interaction happens as well with SIRT1, but not with the others, SIRT2, 3, 6, and 7, which have actually a rather conserved catalytic domain. So quite interesting how sensitive the interaction appears to be to changes in the structure of the protein. So all the work that I'm showing you was conducted by Marius Walter, a senior postdoc in the lab. And one of the other things that he did also in collaboration with Ig-Jung is to look at the subcellular localization of NSP14. It appears to be a whole cell. And you can see with the predominant signal in the cytoplasm, you can see that if we overlap this with the SIRT1 signal, which is almost exclusively nuclear, but shuttles in and out of the cytoplasm, or with SIRT5, we see the strongest colocalization with a SIRT5 signal, suggesting that this interaction might be more relevant, although we found SIRT1 also co-immunoprecipitating. The next observation that Marius made was to show that a single point mutation in SIRT5 within the catalytic domain of SIRT5 is sufficient to abrogate the interaction. This is shown here. So we're looking at two different forms of SIRT5, which are overexpressed here, and you can see a single point mutation in the catalytic domain abrogates the interaction. We see the same thing with SIRT1, catalytically inactive mutant. This is the same histidine residue, which we know abrogates enzymatic activity. Again, here, the single mutation abrogates the interaction. Again, this speaks to a pretty stable interaction. It's robust in immunoprecipitation, and that is also highly sensitive to subtle disruption in the structure of the enzyme. This also suggested that SIRT1 and SIRT5 might be interacting with NSP14 via its catalytic domain. And so the next experiment we did, that Marius did, was to test the effect of a SIRT1 inhibitors that directly targets the catalytic pocket, and this is called EX527. IC50 on the enzyme is around 5 micromolar, and we were delighted to see that at around the same concentration, between 1 and 10 micromolar, we can completely disrupt the SIRT1 NSP14 interaction. And we're currently conducting the same experiment with the selective SIRT5 inhibitor. So this again really points to a very close and high affinity interaction between the sirtuins and NSP14 via the catalytic activity, catalytic pockets of sirtuins. Now, the last observation that Marius made, which is actually quite exciting, is the fact that the interaction between sirtuins and NSP14 seems to be exclusive. That is, if sirtuins are interacting with NSP14, NSP10 cannot interact. And so this is, without going into the whole detail of this experiment, you can see here, this is the interaction between SIRT5 and NSP14. If you overexpress on top of it NSP10, you see a decrease in the interaction. And so Marius explored this a bit further by doing a dose response of NSP10. So if you titrate in more NSP10, you can see the interaction with SIRT5 progressively decreasing. So this suggests a model of a mutually exclusive interaction, suggesting that SIRTuins might be able to disrupt the interaction between NSP10 and NSP14, and therefore might be able actually to act as restriction factors. And we are currently exploring a lot of these possibilities in collaboration with Melanie Ott and the virology group. Some of the key questions that we're asking is SIRT5 a restriction factor, or SIRT1 as well, for SARS-CoV-2 by looking at its expression in permissive and non-permissive cells, by overexpressing it in cells that are permissive, or suppressing it in cells that are non-permissive, or using the small molecule inhibitors. What is the structure of SIRT5 bound to NSP14? As I said, the structure of an NSP14 of CoV1 is existing, and so we're working actually closely with members of this group to generate, to get the SIRT5 and NSP14 interaction. We're also wondering what is the role of this interaction during a normal infection. What is the effect of small molecule inhibitors, either for SIRT1, SIRT5, or both, on SARS-CoV-2 infection? We would predict them to be actually permissive, increasing permissivity. Is the NSP14 SIRT5 complex catalytically inactive as an exonuclease or as a deacylase? And finally, we're very interested in exploring what is the role of protein succinylation, methylation, and glutyrylation in SARS-CoV-2 infection. So on this, I will stop to acknowledge the rest of this amazing group. I don't think we could ever have done all this work so quickly without the collaborative spirit that permeates this, this collaboration. And I very much look forward to continuing our interaction. Thank you.
Thanks, Eric and Natalia. Fantastic stuff. And we will segue immediately to our last speaker, Melanie Ott, who is leading the virology subgroup of QCRG.
Okay, can you see my screen?
Yes.
Okay, wonderful. Hello, everybody. Thanks for sticking around until the last talk. I'm going to represent work from the QCRG virology subgroup. And we in the subgroup are interested in the entire viral life cycle, starting with the entry of the virus through the ACE2 receptor, translation of the 29 viral proteins, interaction with the 332 host factors, formation of RNA replication complexes in the cell, packaging of the RNA into newly formed virions, and assembly and budding of these virions out of the cell. Our goal is to provide the community with useful tools to study each step of this viral life cycle. We're starting with the full-length infectious virus, which is basically mimicking or reproducing all of the steps. We include pseudotype viruses where we just use the spike protein and put it onto other viruses like lentiviruses or VSV to just mimic the entry step of the virus into cells. We maintain interest in the individual viral proteins and subgenomic replicon clones, which are focusing on the RNA replication step in the virus where you just have positive strand RNA replicated via negative strand intermediate back into positive strand RNA, but without production of the full infectious virions. We are also interested in the production of the virions by producing virus-like particles where we keep out the non-structural proteins, but just focus on the structural proteins to just see whether they form by themselves viral particles that can be used to query this final step of the viral life cycle. And also, we're in the process of setting up animal models that can be used to test therapeutics in the future. So my lab has been working together with Anita Seal's lab at the BSL-3, airborne pathogen BSL-3 in Parnassus to culture the virus and do first infection experiments. We hope to move to our facility at Gladstone in the next week or two. And we have so far infected about 16 plus different cell types, including transformed cell lines, like here the CALU3 cells, or more sophisticated cell models like iPSC-derived cardiomyocytes, together with the cardio group, Todd McDevitt and Bruce Conklin. Or my lab is involved in generating and infecting adult stem cell-derived organoids, including lung and intestinal organoids. And here on the top, you see the mock-infected cells, and on the bottom, the infected cells. And we're staining here for double-stranded RNA, which is selectively picking up replicating positive and negative strand RNA replication centers in the cell, and in our hand is the most sensitive and specific way to detect the virus in the cell. And you can see CALUs here in green are massively infected. You see infected replication centers everywhere. Cardiomyocytes also extremely well infected with beautiful replication centers in the cytoplasm. In contrast, the lung organoids are much less hard to infect, and I think we think that this is because our airway has been exposed to coronaviruses and have evolved to sort of protect themselves from them in a more sophisticated manner. And so we're working very hard in increasing the so far low infection rate that we see in these. I'm showing you here the lung organoids on top, and in green the ACE2 receptor, because you can see how delicately and how precisely these receptors are expressed on these more complex cell models. You see that they are polarized strictly to the apical side, which is exactly where we want to have them, because that's the luminal side from the gut. We have also established a pipeline to test therapeutics. We heard a lot about interferons. Interferons are really very powerful antivirals. There was a little bit of confusion in the field because a Cell paper came out saying that interferons upregulate ACE2 and might therefore promote infection, overriding the antiviral effect that we see with interferons, but this is not what we see at all. If you treat virus cells that replicate the virus with interferon alpha or interferon beta, you see that you completely block replication. And this is abrogated, this block, and when you treat the cells also with a JAK inhibitor. You saw from John's talk that JAKs are important signaling mediators of the interferon response. And so if you block the signaling of the interferon response, you restore infection or you even increase it. Other inhibitors like ribavirin, which is sort of a pan-viral inhibitor, don't work very well in these SARS-CoV-2 infected cultures. We're measuring here RT-PCR of either the E gene or the N gene in the supernatant of the infected cultures. Our focus maintained is still very much on the 332 host factors. Dave keeps us all focused on them. He and Joe Hyatt are focusing on, establishing a high throughput CRISPR knockdown pipeline in sophisticated cell models, such as the primary bronchial epithelial cells. They basically nucleoaffect them with the preformed RNPs, Cas9 guide RNA RNPs, to establish polyclonal knockout cells. And then when you seed these cells in a transwell and put them at the air-liquid interface, you can really very strongly differentiate that so that they upregulate the ACE2 receptor and can become permissive. And we are currently infecting these cells together with Dave. He and potentially also his knockdown cells in the very near future, because he and Joe have really done an exhaustive analysis of buffer and different conditions that they can use to enhance their transfection efficiency or editing efficiency up to 80% in these very complicated cell lines. I told you that one of our focuses is on the RNA replication centers to generate subgenomic viral clones, which can be used at BSL-2, which do not produce the full length, or the infectious virions, but just reproduce the RNA replication step in the infected cell. And this is something that Emily and Beatrice and Charlie's lab have taken on very early by reproducing basically a clone that spans the almost 30,000 nucleotides of the SARS-CoV-2 viral genome. And here, in order to generate a non-replicating virus, they basically omitted most of the structural proteins here, excluding the nucleocapsid, and have replaced it with either reporter or selection markers to generate a stable cell line that would autonomously replicate the viral RNA in the cells. So you can see so far they have been successful in generating, the DNA template by ligating different G blocks corresponding to the different parts of the genome together. They have also succeeded in generating high content RNA transcription, in vitro transcribed RNA of this construct, to then go on to transfections and selections in cells, which is currently ongoing. And I know they're very eager to see whether some of these transfections have yielded into a replication, autonomously replicating clone. Now, if we focus on the other side of the equation, not the non-structural side, but really the structural side or the late stage of infection, and then we basically want to omit all this, what I'm just telling you, RNA, non-structural proteins, and just purely focus on the structural proteins, and whether by themselves, if you transfect them in themselves into the cell, would form virus-like particles, which are basically empty shells without a viral genome in it, but which would reproduce very precisely sort of the morphology of the real virus. This has been taken on by Mali Safari and Alan Franken's lab, because on the premise that for SARS-CoV-1, this has been successfully done. If you co-transfect the M, E, S, and N proteins together into different cell types, you can indeed generate these virus-like particles that you can see in electron microscopy. And so Mali has so far successfully expressed these different proteins in cells, and is currently optimizing the co-expression experiments to then concentrate the supernatant, harvest the VLPs on a sucrose gradient, and examine whether she really has virus-like particles in the supernatant using electron microscopy. Finally, I think there's still a lot to be done just using the 29 viral proteins that we have of SARS-CoV-2, and especially comparing it to the other, the proteins that are encoded by SARS-CoV-1 and MERS. Jen and Chris in my lab have been playing around with a new high throughput confocal microscope at Gladstone, where they specifically asked questions how they could use that microscope to query function or functional changes that they could see in these transfected cell lines. And here they're focusing on phosphatidylcholine, which is a major component of cell membranes. And they're testing using click chemistry, using a fluorescent dye, how expression of viral proteins would affect the synthesis of phosphatidylcholine. Now, phosphatidylcholine is important for viruses because viruses like SARS-CoV-2 produce high amounts of intracellular membranes to protect their RNA replication centers, so that they cannot, the RNA that's being produced is not detected and sensed, but it's also not degraded. And since PC, phosphatidylcholine, is a major component of these of membranes, we think that we can probably derive some sense of how viral proteins are affecting PC synthesis, and how this might correlate with the generation of these membranous webs, which is a critical factor. And you can see here that we see that some of the proteins, including NSP10 and 15, but also E and ORF8, have increased PC synthesis in just stably expressing A549 cells. And then you can compare whether that's the case also in the other viruses. So in conclusion, I think virology is hoping to establish tools that will help all of the subgroups here at QCRG. We have, intense interactions with all of you, but specifically with translation, mitochondria, metabolism, and epigenetics in terms of focusing on individual proteins, and with cardiovascular with infecting the iPSC-derived cardiomyocytes. But we look forward to work with all of you. With this, I end. This is the amazing group. I thank you all for your hard work on this, and thank you for your attention, and I think we're ready for questions.
Thanks, Melanie. Such fantastic tools that'll have huge impacts across the board and all the subgroups. So we have a couple of minutes. I see that John and Natalia and Danica were dutifully typing in their answers to a number of questions. So that's great. Thanks, guys. So maybe just a few we can highlight here. One for you, Melanie. Can these VLPs, can they reinfect other cells?
No, they can. So they can be taken up, but they cannot be. They are not infectious. But they can bind, and they can be maybe engulfed by macrophages, but they are non-infectious.
And Eric, maybe one quick one for you. Does this interaction between NSP14 and SIRT5, does it occur when other components of RNA polymerase are present?
It's a good question. We haven't done this. We're planning to do this in collaboration with Melanie using the replicon.
Maybe one more question here from Kayvon. It's kind of a general question. Maybe, I don't know who would want to answer. Maybe this is for Melanie, but the question about combinatorials. So obviously remdesivir is the one drug that's being used. I mean, what are your thoughts in terms of what could be used in the future? What is your gut telling you, Melanie?
Well, we haven't tested remdesivir very extensively so far. I think the effects are not very strong in vivo. I think and as we all know, I think using one antiviral will generate resistance in potentially no time, which we want to avoid. I think we can go. I think there's different avenues that can be pursued here. I think we can treat the cells with remdesivir and combine it with a CRISPR screen and see what pathway in addition would be interesting, or we can do combinatorial screening, or we can do candidate screening. I think both Nevan and I, we are extremely fond on host targeting the host to avoid resistance and to have a broader effect. And I think a smart combination between an direct acting antiviral and potentially a host targeting antiviral would, in my mind, go a long way.
All right, that's great. So we have 20 seconds left. And so I just wanted to thank everybody for presenting such great science, such great collaborative science. It's just so exciting to see all the crosstalk between all the different groups. And I'm not sure if all the participants can see how many people were on listening, but just to tell you, it was the highest I saw was 192. So that was a great turnout. Thank you for listening. And we've recorded this, and this link will be on the QCRG website, as well as I assume on YouTube as well. And the next town hall we'll have the last 4 subgroups present. So thanks everybody for participating, and we'll see you all soon.